our soil contamination and remediation course. I hope you're enjoying the lectures if you have been listening so far. If you're new, please feel free to ask questions. You can do this by typing through the chat box, or you can wait to the end of the lecture and ask questions verbally or type them in then as well. You can stay on also for discussion with Hui after the lecture is finished. I would like to let you know of a change to the order of the lectures in the weeks that are following on from this one. So now lecture 5, that's next week, will be my lecture on remediation of chromium contaminated sites. And then in week 6, it will be Huan Zhong from Nanjing University, followed by Professor Ji from Nanjing in week 7, and then week 8 will now be Professor Ma from University of Florida. So that's a change, and that information should also be on the IIES website. So now it's my great pleasure to introduce Rainer Schulin. He's a Professor of Soil Protection and indeed Head of Group in the Institute of Terrestrial Ecology at ETH Zurich. During his career, he has studied a wide range of subjects. He has degrees in biology and separately in zoology. He gained a doctorate in zoology before undertaking a further course of study in forest sciences and graduated as a forest engineer. He then had a period of research in soil physics before taking up his current position as Professor of Soil Protection, where his main focus is on assessment, analysis, prevention and remediation of soil degradation by heavy metals and also by compaction due to mechanical stress from heavy machinery. He's authored more than 200 peer-reviewed journal articles. He has co-authored textbooks such as a textbook on soil ecology. He's been a scientific expert in many German and Swiss science foundation panels. He's on the executive board of the International Society of Trace Element Biogeochemistry, and indeed he is the chair for the forthcoming 14th International Conference on the Biogeochemistry of Trace Elements, which runs this summer, July 16th to 20th. Maybe some of you will be going to that meeting in Zurich. And today he's going to share with you his work on shooting range and military sites in relation to antimony behaviour and its potential impact on human health.
Thank you very much, Margaret, for your nice introduction. And welcome everybody to this lecture today. It's the first for me on video lecture, and I hope you can understand me. So let's start the presentation. Can you see it? Okay, good. So shooting range soils is an environmental problem in Switzerland since many years. At first, it was thought to be mainly a lead problem, but in the last 2 decades, it was increasingly becoming clear that there is also a problem potentially with antimony. And so we started working on that problem, let's say about 10 years ago. And what I'm telling you today, that is heavily cannibalizing on the work of my former PhD student, Kerstin Hockmann, who is now a postdoc in Australia at the Southern Cross, and Susan Tandy, who is my senior researcher and who supervised also this work. And most of the material I'm showing you are actually their work, and I just put it a bit together and presenting it now. Okay, I start with what is antimony. As I said, antimony is not that long in the environmental agenda. It is an element that belongs to the group where we also find arsenic and phosphorus and nitrogen. And for a long time, it was thought that antimony is pretty similar in its environmental behavior to arsenic. But this is partially true, but not completely. Arsenic is like antimony, not a real metal. Antimony is more metallic than arsenic. It is a brittle element, and it has like arsenic mixed metallic and non-metallic properties. Antimony has, as I said, some properties that are similar to arsenic, especially the redox behavior. I come to that in a minute. While the deprotonation, the acid-base behavior is quite different. It exists in two main forms or oxidation states in the environment, like arsenic. Under oxygenated or aerobic conditions, in solution, it is primarily present as so-called antimonate, which is Sp5, oxidation state 5, OH6 minus, means it is an anion, while under reduced or oxygen deficient conditions, it speciates as antimonite. So Sp3, as antimony with oxidation state 3, and that speciates as SpOH3 as a neutral molecule. And here you see the speciation diagram in the x-axis pH and in the y-axis the redox potential in volt. And you see that above this line we have the Sp5 form, the antimonate, while below antimony is primarily or is speciating as antimonite. And you see also that pH over the range, which is for environmental conditions or soil conditions typical, it is present always in this state as antimonate under oxygen conditions and antimonite in the reduced conditions. So these different protonation and deprotonation state, they occur at pH values which are not relevant under normal environmental conditions. And this is a difference to arsenic. For a long time, it was not really clear the condition or the function that antimony has or the effects on biological tissues or organisms. This has to do with the fact that it was difficult to distinguish analytically antimony from arsenic. And so there was always some uncertainty to what degree antimony effects were actually caused by impurities of arsenic. But it is well known that like arsenic, antimony has no essential biological functions, but at high concentration, it is also known that it is toxic to plants, to animals, and humans. In particular, it was also used in the form of potassium antimony tartrate to treat humans against parasitic diseases, in particular leishmaniasis and also schistosomiasis. I hope I pronounce this so you could understand it. It is also has been found that the reduced state of antimony 3 is more toxic than antimony 5. And it is suspected to be a carcinogen. Antimony has been used already in the ancient times to the old Egyptians and was used as eyeliner. And in later times, as I just said, it was used as a medicine. It was also used by doctors to make people vomit in order to empty their stomach, so as an emeticum. And there are rumors, they suspect that the antimony was a poison that killed Mozart. Antimony now has many other uses, so it is used as a flame retardant. For example, it is part of brake liners in cars. It is used in plastic synthesis as a catalyst, as a decolorizer in glass, and especially also it's used as a component of metal alloys. And this is a use where we will later see is problematic in shooting because it is used to harden lead. And there are many other uses. And there's, no, I need to, why doesn't it move on? Oh. And so there's no wonder that the consumption or the use of antimony has increased a lot in the last few decades. Here you see statistics from the US Geological Survey, which shows that we had a steady increase until 1970, 1980s, and then a very rapid increase during the last 3 decades. And so it's no wonder that we have also increasing release of antimony into the environment, at first through the mining processes, then also with the incineration of wastes that continue and that contain antimony. Then, for example, through traffic emissions, as I said, these are primarily due to the brake liners. So crossroads, wherever there are people brake their cars, there is also increased antimony concentrations on the roadside soils. And there's also antimony in fossil fuels, which gets emitted with the incineration or the burning. And last not least, or for our purposes here, it is the release of antimony with bullets in shooting ranges. And this is, as I said, the main problem at the moment related to antimony in Switzerland, but also in some other countries like Norway or the US, which have a lot of shooting apparently. In Switzerland, this is an issue because, as you may know, Switzerland has a militia system, and the soldiers or the militiamen are requested, we also have some militia women, but the majority is men, are requested to shoot every year to get a certain, to get practice, so they have to shoot and get a certain number of points. And for that, there are thousands of small shooting ranges all over the country. There used to be more than 4,000, many have been closed in the meantime. There are still more than 2,000 active, and these shooting ranges usually have a size of 300-meter length. So at one end people shoot, and at the other there's a stop butt where the ammunition is caught. So in total, it is estimated that more than 2 billion projectiles have ended up in these soils around these stop butts primarily. And if you consider that the bullet contains 1 to 5% antimony as lead hardener, in average around 2%, then this amounts to quite a bit of input. In the soil, the antimony or the bullets generally corrode very quickly within a few months, as you can see here in the upper right. And then the metals are released, and also the antimony, in the surrounding soil, and it gets oxidized from the metallic form to either antimony 3 if we have anaerobic conditions, or even further to antimony 5 if we have aerobic conditions. So some estimate of the total amount of antimony that ended up so far in Swiss shooting range soils. There are estimates that around 430 tons of ammunition is in these soils. The annual input is estimated to be about 10 tons or more. There are differences in the estimates. And that the total stock is around 2,600 tons. So although it's locally concentrated, it's spread all over the country due to the large number of shooting ranges, and there are potential problems with environmental and human health risks. For example, one pathway is that these shooting ranges are often used for pasture when there is no shooting activity, or after the ranges have been decommissioned and they are then used for agricultural purposes. Not only pasture, but also sometimes for crop production. And some ranges are also located in wetlands. So here you see some limits. There are stopped again. Why is that? Forward. Okay. There is a limit of 5 microgram liter in drinking water by the EU. In Switzerland, the only value that relates to waters is a value of 100 microgram per liter in discharge water, so from water treatment plants or other water that is collected, rainfall runoff. It needs to be below this 100 microgram liter antimony to fulfill the legal standard. There's also value now for soils in children's playgrounds. There is it's rather high actually. 50 milligram per kilogram antimony is permitted. And I put, actually Susan put together here values from other countries where we find values between 10 and 50 milligram per kilogram soil. Okay, so the question is what we can do with the soils. Because if we would dispose of, one option is disposing on those disposal sites. But if we would dispose of all these large areas in total fitting range, it will amount to huge volumes, and the costs are quite high. Another option is soil washing, but soil washing is not feasible with all types of soil and also is not cheap. So one option that was and is considered for soil that is not heavily, but let's say intermediate, is contaminated in an intermediate range, is to reuse it for non-sensitive purposes. For example, to refill in landscaping roadsides, so where we anyway have already contamination. And this is the option where we were then looking for what kind of risk do come from this reuse. And the risks are in two ways. One is the food chain, which I'm not talking about today. And the other is leaching with water, infiltrating water and leaching into groundwater or surface waters. So the question is, what is the potential of mobilization and leaching? And for that purpose, we constructed a Lysimeter facility to investigate this problem under real world conditions. And you see here the scheme of this facility. We have 8 basins, and they're shown here. Each basin has a surface area of 5 meter length by 3.5 meter width, which is close to 18 square meters. And we have a depth of 70 centimeters of the soil above a drainage layer. The outflow is collected and sampled in a separate room, and so we are measuring the total discharge as well as the concentration, but we also measure other things. Here you see some pictures of how these lysimeters were constructed. Here you see one of the basins. Here you see the tubing, slitted plastic tubings through which the water can be collected and is drained to that room. On top of it, then we have the gravel coarse sand layer that drains the soil. On top of this 20 to 30 centimeters drainage layer, we have then the soil, 70 centimeters of soil. And all these lysimeters were then seeded with a pasture, a mixture of grass and ley, which is used by farmers for producing feed for their cattle in Switzerland. It's a standard mixture. You also see that we have here a weather station, so we monitor rainfall and so on and so on. And the two soils, they come from 2 shooting ranges that have been decommissioned and remediated. One of these sites is in southern Switzerland. Just to give you the geography, you see here Zurich, here would be the Alps, and this is south of the Alps. It's bordering to Italy, and there is a military site with a shooting range at the edge of a wetland. And just see in a minute the properties of the soil. And on the other soil was from the river Rhine Valley of eastern Switzerland near the city of Chur, where we have alluvial side, so with sandy, silty soil, which was used actually during the remediation, which was used for production of some agricultural crops, including potatoes. And here are the soil properties of these two soils. So the pH was slightly acidic in the side from Lysimeter, the southern Switzerland side, and around 8 in water in the Chur soil. This was due to the fact that we have calcium carbonate in this material, so about 15%, while there was no carbonate in the Lysimeter soil. And on the other hand, we have quite a different also in the organic matter content. So organic carbon was very low in this alluvial soil, 0.8%, while it was more than 5% in the southern Swiss soil. The lead concentration was similar. It was around 500 to 600 in these soils. And also the antimony concentration was similar. It was around 20 ppm in those of these soils. Okay, here you see now the discharge composition during our first few years. I put in here the limit in Switzerland of this 100 microgram per liter for discharge water and 5 milligram per liter limit of the EU drinking water standard. And you see the squares that give you the effluent concentrations of antimony, of total antimony from the Chur soil, and triangles from the Lysimeter soil. And you see there's a little bit more leaching from the soil with less humic matter and higher pH. And you see that we are partially exceeding here the limit for water discharge. So this actually we would not be allowed to run these lysimeters legally without cleaning the water. But fortunately, it was only during a short time, and so we did not need to install water treatment. But it would certainly not be allowed to drink this water, to use it as drinking water. Okay, why do we have more leachate, higher leachate concentration in the Chur soil? Does not work. This has to do with the absorption of antimony to the solid phase. And here are some values or some data from the literature. Here you see, unfortunately, it's a bit confusing. I always have to orient myself on this. You see here two different scales. On the right-hand side for antimony 3 micromole per gram, and from 0 to 12, and on the left-hand side for antimony 5, but it's times 10 the concentration. But unfortunately, the dark symbols, which are more on the left-hand side, so relate to this scale, while the open symbols relate to this scale, as you can see here. And it's for two clay minerals, montmorillonite at pH of 7.3, and for kaolinite at the pH of 7.9. So at the slightly basic or alkaline range. And what we see is if you relate this, they are not so different, the two antimony species at the especially at the lower pH of montmorillonite. We see would see a little bit larger difference for the kaolinite. And what we see is, or what we do not see, sorry, what we could see if we would have the data on the same scale for different pH, that we have lower absorption for the higher pH. But this comes clearer here. I show you data for antimony 5 adsorption to goethite. And absorption to the iron oxides is actually much stronger than to clay minerals. So this is what really is important. And here you see a comparison of the absorption of antimonite made supplied at a concentration of 4.1.5 micromolar at different pHs, and compared for two ionic strength, IIES ionic strength of 0.01 molar, and the open symbols for 0.1 molar, so you see there's a difference. But also it's compared here to phosphate sorption and to arsenate sorption. So this is arsenate and this is phosphate. And we see that at high pH, there's quite a difference. The antimonate becomes less and less strongly adsorbed in contrast to the other two solutes. And this is due to the fact that we have only anions, and that the higher the pH is, also the more negatively are charged are the surfaces of the iron oxide, and so we get less strong sorption. And these are the two pHs for the two soils. And there you see or the difference for the two soils we have, and this explains, I would say, to a large degree, the difference we found in the discharge. And just to remind, the binding strength of iron oxides is about 10 to, depending on pH, 10 to 100 times more than to iron oxide to clay minerals. So we neglected clay minerals. Okay. Before I continue with the lysimeter experiments, I show you now some work Kerstin did on mechanisms. We were particularly interested in the behavior of the soils under reducing conditions, because, as I said, many of these soils are wetlands, former wetlands, or also pasture soils in Switzerland are often waterlogged, so the change in redox potential often plays an important role. And there was very little knowledge 10 years ago, at least, about what effect would that have on the mobility of arsenic in such soils, antimony in such soils. And we started, or Kerstin started with microcosm experiments. All these experiments now are performed on poor soil. So the soil with the higher pH and the lower organic matter content. That's also the reason why I didn't go into too much until now into sorption by organic matter. And she performed treatments with and without addition of sodium lactate as a substrate for the microbiology in order to stimulate them or not to stimulate them. And the microcosms were purged with molecular nitrogen in the air, so to remove the oxygen and to keep them under, so to generate or to produce anaerobic conditions through the consumption and the oxidation of the lactate. And in some samples, half of the samples, Kerstin reopened them after 6 weeks, so to re-oxygenate them and look at how that would affect the solubility. And all these experiments were buffered, and the pH varied time a bit, and was kept as good as possible between 7.3 and 7.8. So I have to go back. I have a problem with myself. I don't see this. Okay, I see it. So the liquid solid ratio was 10, so 5 grams per sample were then diluted or mixed with 50 milliliter of solution. As I said, we had several weeks, so up to 64 weeks. This is the lactate, and the antimony was analyzed with HPLC ICP-MS, the speciate between SB5 and SB3. Whenever I do. Now, here you see what came out. This is the time axis, so up to the 64 days, more or less every 7-8 days a sample was taken, or 3 samples were taken for each treatment. And sometimes they had a little bit more variability, as you see here, but mostly the error bars were within the size of the symbol. And the dark symbols relate to the non-oxygenated samples. And after, as I said, 42 days, 6 weeks, some were re-aerated, and these are the open symbols. And you see that the redox potential quickly dropped within the first few days to around 0, and then also very quickly after re-aeration, it increased again to around 400 millimoles. The effect this had on the antimonate, so the SP5 in solution, is shown here. You see that the antimonate concentration equally rapidly decreased as the redox potential decreased. And we also found that it increased again as soon as the samples were re-aerated. What happened to SP3, antimonite? Interestingly, also the antimonite increased, but more slowly. Well, we should have expected, actually, if there would have been no sorption, that it would increase rapidly, right? If everything would have remained in solution, the missing antimony 5 here should have been exchanged against antimony 3. We don't see that. But the antimony 3 still increases more slowly and then has a tendency slightly to drop or remain stable. And as soon as it's re-aerated, it drops again in opposition to the SP5. So there we see that the SP3 is oxygenated, is oxidized, and reappears. But there is something missing. So if we, as I said, if we add these two curves, we would not get this constant amount of antimony. So where is the rest? The rest apparently must have been sorbed. But why does it reappear? That has nothing to do with the redox potential. The clue we got when we analyzed the analyzed, I should say this, the iron in the solution, the iron 2. And here you see that there's quite a parallel increase in iron 2. So this suggests that there were iron oxides that got dissolved due to the reduction of iron 3 in the iron hydroxides and oxides to iron 2. And the iron also got rapidly removed again with oxidation and precipitation. And with that, also the antimony 3 that was left was sorbed again. The amount that was not oxidized to antimony 5. Here you see the speciation diagram for antimony together with iron for the conditions we had in our batches. And forget about here there's a sulfur, which we did not have in our system, but just concentrate on the upper part of this diagram. And you see in the green line gives you the separation or the dividing line between iron oxides, so oxidized iron and iron 2 plus in solution. And we see that, you can see at the pH that we had in our system, antimony was reduced or should have been reduced already at the redox potential higher than the iron reduction occurs. So that explains why we had first the reduction and production of antimony 3, and then only when the redox potential decreased further, where this reductive dissolution of the iron oxide started. And I showed you before the sorption pH dependence for the antimonate here. And here I show you the same diagram now for the antimonite, a little bit lower concentration of 2.2 micromolar. And what you see here is that we have no dependence on pH least for that pH range that is environmentally interesting. And if you compare now the effect at the pH that we had in these soil solutions, then this explains why the antimony 5 was more soluble than the antimony 3. So with the reduction of the antimonate, we had to expect that the product, the antimonite, the antimony 3, was stronger sorbed and thus disappeared from the solution. Okay, to test a bit more this hypothesis, Kerstin performed additional experiments. She performed the same experiment also without adding lactate. These are the results you just saw. We put them into the same graph for antimony 5 and antimony 3 and only show you the results with re-aeration. And here you see the iron. And now these are the results. These are results for experiment without sodium lactate. We did not sterilize this soils. So there was the autochthonous microflora. So we had also consumption of the substrate that was in the soil, and that led to the decrease of over time of the redox potential. I have not shown it here, but it just occurred much more slowly and more or less in parallel to the decrease you see here in antimony 5. What was interesting to see that we have a peak here. This was not evident in the experiment with the lactate. And I'll come to that later, what happens here. Why did the antimonate concentration increase? We had no change in the iron solution during that phase. There was a little increase here that may have been due to inhomogeneity of the soil. These are the same two graphs that show you the relation between antimony and the iron. Our suspicion was there are also other hydroxides and oxides in soil. And one important component are manganese oxides. They are quantitatively much less important than iron oxides in our soils, most soils I would say, but still they can play a role and they act as sorbents for antimony like iron oxides do. And you see that the manganese, the free manganese concentration in the solution increased much earlier than the iron, which you would expect, because manganese is reduced at much higher redox potentials already. And you see here that more or less in parallel, when the antimony 5 concentration increased, we also had this increase in manganese. So our hypothesis is that it was a similar process as with the iron. We saw it in the lactate experiment that reductive dissolution of manganese oxides led to the release of antimony 5 that was sorbed before to the manganese hydroxides and oxides. To test the hypothesis that there was really redox changes in the antimony speciation, Kerstin also performed potassium etch synchrotron X-ray absorption near etch spectroscopy of the antimony on the soil in these experiments after the microcosm experiments have been finished. Well, and on the initial soil. So on the soil that was reduced after some weeks and the soil before it was put in. And here are some pure substances for calibration, for comparison: metallic antimony, antimonite, solid antimonite, and then produced antimony 3 sorbed on goethite, antimony 5 on goethite, so on pure mineral, potassium antimonate and calcium antimonate as potential also phases of antimonate precipitation in these soils. And with a linear combination of the functions that describe these curves, Kerstin fitted the curves that were then recorded for the initial soil and the reduced soil. And with that, she was able to come to an approximate quantification of the redox species in these soils. So in the initial soil, we still had some reduced antimony, but the majority, almost 90%, were antimonate. And the opposite was the case after the reduction in the anaerobic conditions. So I'll summarize our results and conclusions from these experiments. So we hypothesize that we have first, when the redox potential decreases, a reduction of the manganese, with that mobilization of antimonate that is bound to the manganese. Then if it drops further, then the antimonate is reduced to antimonite. And when the redox potential decreases even further, which is sorbed, on the iron oxides, and this further drop, when these get dissolved by reduction, we get again mobilization, but now of antimony 3. In order to also to check this under flow conditions and get better evidence, Kerstin performed also column experiments run under saturated flow. These columns were also used with an influent solution of sodium lactate, and the rate of the flow was around 6 pore volumes per day. So this is a very high rate compared to environmental conditions. And in order to look for conditions also of non-equilibrium kinetics, there was a flow interruption between day 5 and day 45. So that is the gray part where we have the interrupted flow, and here you see the effluent concentration of total antimony and the percentage of antimony 3 in this effluent. And you clearly see that in the beginning, sorry, this is for the sterilized columns. Here we use sterilized soil and compared it to non-sterilized soil. And the sterilized soil, which was sterilized with gamma ray irradiation, showed initially the same behavior as the non-sterilized. So this is not the beginning is not due to a difference or to any microbial effect. While we have the second peak here in the untreated soil, obviously had to do something with the sterilization. Also, the change of concentration of that occurred during the time of the stop flow, and after it was continued afterwards, where we see that we already see here a slight increase in the antimony 3, and we had a high percentage of antimony 3 after these 42 days of, of these 33 days of first flow stop. And again, what is the reason for this? After our batch experiments with the microcosm, we suspected that this was due to manganese, but before I come, sorry, this is shown here. Here you see the manganese concentration in the effluent, and here you see the iron concentration in the effluent, and this is confirming our results of the batch. This second peak really emerged when also manganese appeared in the solution. So this is in agreement with this hypothesis. This initial peak and also this increase after the flow was resumed indicates that we have some maybe physical, maybe chemical non-equilibrium effect with desorption, sorption, or exchange with less mobile phases. What happens during this flow interruption phase is shown in this graph. So we also see here there is some non-equilibrium effect, but then the antimony concentrations remained stable. They were higher in the sterilized soil, in agreement with the fact that we have here more reduction, while little happened in the untreated soil. Especially we see this in the Sp3 concentration, and we see this in the manganese concentration, and we see that manganese also increased slightly during that phase. So apparently the columns did not completely remain sterile over the entire time of the experiment. But we do not see iron reduction in the sterilized columns, only here in the untreated, so in the non-sterile columns. To check further this hypothesis that manganese oxides and hydroxides are really also important as sorbing phases, here are some results from the material taken from these columns and analyzed by elemental laser ablation mapping. And you see here 4 patches, A, B, C, D, in all these pictures. And you see here the various elements, antimony, iron, manganese, lead, copper, and calcium, which were analyzed for. And if you see, compare, for example, in A, we have antimony collocated with manganese, but not with iron. While in B, we have the opposite. It's collocated with iron, but not manganese. We have also lead and copper, which indicates that this was from the bullet weathering. The bullets also have a copper mantle. And B and C, C and D, they show both elements, manganese and iron with antimony, and also in case of B, we see again lead and copper. So in the case where we have iron oxide absorption, it seems to be related to where the bullets came in. Maybe the iron actually also was the rest from the steel mantle and then oxidized, while the antimony and the manganese was from antimony that then migrated and was sorbed and trapped. Conclusions of the column experiments. So our end of the batch. The manganese hydroxides can be important sorbents of antimonate. The antimonate released, or antimony 5 released from manganese hydroxide solid phase through reductive dissolution is reduced then to Sp3 with further decrease in redox potential. And we find substantial solubilization of iron, antimony 3 with reductive dissolution of iron hydroxides in the biological active soil. So, and at the last part of my talk, I come back to the Lysimeter experiments. Now we were, of course, interested how this would play out in the field. Sorry. Okay, here see the Lysimeters after an initial phase where we run them all with normal drainage. We wanted to see how waterlogging would affect the dynamics of the antimony in the system. So we installed a valve, and the valve stopped the outflow when the water level decreased below 50 centimeters depth. And it opened again when the water level increased above 30 centimeters. So we tried to keep it in this range, simulating the behavior of so-called groundwater influence or glycol, which often occur under these conditions in this pasture land and especially also in wetlands. And now I show you the first the redox potential, how it responded to the treatment. The top graph for the control and for the lower graph for the waterlogging treatment. Here you see the redox potential measured at 4 different depths, 10 centimeters below soil surface, 20, 37, and 54. So that was just a little bit above the centimeters, 70 centimeters was the entire depth, but it varied, so it was a bit the lowest measurement we could take above the drainage layer. And you see that actually already under normal conditions, the redox potential dropped in the growing season and then increasing over the winter, the non-growing season again. And this is due, and even in we get some decrease in the upper soil parts horizons in this soil under conditions we consider normally aerated. And this happened in all 3 years you can see here. So apparently the exudates of plants primarily, which enter the soil, get consumed by the rhizosphere microorganisms primarily, microorganisms in general in these soils, And this leads to a lack of oxygen and leads to, in these lower depths, to phases of anaerobic conditions. Here you see the same or analogous data for the waterlogging treatment before the waterlogging was implemented. You see also how well or how non-well the two lysimeters, in each case we had two of these lysimeters. Remember again, these were only done with poor soil, so we had two with waterlogging and two without waterlogging. And of course, you always have some spatial variability. The packing is never really the same, so there is a little difference here that actually this depth 37 was better aerated in the waterlogging treatment before the waterlogging than in the other lysimeters. But otherwise it was the same. So the difference now comes here after we started in mid-2012 to log the water. It took 6 weeks until the water, because it was happened to fall into a dry period, unfortunately. So we had to wait 1.5 months, almost 2 months, until we got the rise in the water table. So can you still hear me? They are working, unfortunately. Okay, so you see what the difference comes next spring when the comes in the next winter when the water potential were supposed to increase again, as I did here. Then they were low, and then they started to decrease also in the upper horizon. So only in the year after we started waterlogging, we really saw a strong effect also in the upper horizons on the redox potential. And how does this now, what consequences did this have on the effluent? Before I come to the effluent, I show you we also took soil solution samples. I show you here two examples for two depths. These are for the depths at 20 centimeters, the open symbols, and at 54 centimeters. That's the closed samples. As to be expected from these redox potential measurements, we had lower antimony 5 concentration in the soil solution below because it was more reduced. Until the waterlogging started, they should have the same, but we saw there were differences, and we have a lot of spatial variability with redox potential, so that's nothing to be really concerned about. But after the waterlogging, here we see that the SP5 concentrations get lower and lower, also in the topsoil, and remained low. They did not in the other soil that was not waterlogged. And we found no, I mean, here we found also some reduction to antimony 3 in the lower parts, but nothing after the waterlogging. And we found slight increase in antimony 3 over time in the lower horizon in the waterlogged soil solution set. In the effluent, all these local heterogeneity evens out, so we have much less difference between the two treatments here when we compare the effluent in the discharge, so the concentrations of antimony, manganese, and iron in the discharge. And here we see that we have nicely the same until the waterlogging started, and then the antimony, the total concentration, decreased as we would have expected from our, and we did expect from our microcosm column experiments. And we also found that manganese concentrations started rather soon, already in the fall-winter of the first year, while the iron concentration only started slowly to increase at the end, but during the second year, especially at the end and in the third year. But we do not see that here there is an increase in antimony, which would be due to the reductive dissolution. Well, the scale. If we change the scale, so to a much higher resolution, then we see, oh yes, there was nonetheless some increase did start. So while it was not far as strong as we observed in the columns or in the micro, we still found this increase, and it was more or less related to this increase in the iron. So we think that this was a main process, and if we correlate the iron concentration in the effluent for this phase after this first drop, we are still in the drop phase, then we found quite a nice correlation, which is in agreement with the hypothesis that it was mainly reductive dissolution of the iron oxides. So I come to the final conclusions. So the leaching of antimony under field conditions was dominated in the Lysimeters by the antimony 5. Also under waterlogged conditions, at least for the 3 years when we observed. We observed until the spring of the year, which is, sorry, we get it until end of 2014. We continued until spring 2015 to have 3 years, but nothing then really changed much more any. And we found some reduction of antimony 5, as I said, to antimony 3 under these conditions, but very slow. And the release of antimony 3 from iron and manganese hydroxides under these field waterlogged conditions did occur, but played a minor role over the 3 years of waterlogging. And with that, I finish my lecture. Thank you very much for your attention, and I'm happy to answer any questions. Thank you. I have a problem now. I don't hear.
Can you hear me now?
Now I can hear you, yes.
Yes, yeah. So thank you very much. That was an excellent presentation, taking us through all the different aspects of your research on antimony and all the different types of experiments. And the first thing I wanted to ask you about was about when you find manganese oxides being important. You showed us the evidence of the association between the antimony and the manganese in your soil profiles. And do we know how this interaction comes about? You have a negatively charged antimonate interacting with a manganese oxide surface. Do we know the mechanism of that interaction at the current time?
Yes, it will be surface complexation, and the manganese oxides have actually a much higher point of charge. So they won't be as negative as the iron oxides are at the around neutral pH. Which is slightly basic pH. So that's what I think is the reason why the manganese sorbs more antimonate than the iron oxides do. Yes,
okay, yep. Do I see another question? As a question here from Ian Oliver. Will it be feasible to keep these sites waterlogged in a management attempt?
You mean as a kind of a remediation option to reduce the leaching of antimony? Is that your question? So that would be
yes, that was the question. If we could reduce the antimony and keep it from coming into solution as a wetland, would that be an option to make it a natural park wetland area, and then problem is solved, or is that not feasible?
Well, when you have this condition, that is probably feasible. Like that one actually where this Lysimeter soil came from, with I thought wetland was a swamp. You could probably just restore the original hydrology, and so be rather sure that the antimony will be reduced. But the problem is you have to make sure that this does not lead to more reduction of the iron, because we observed it over 3 years there in these lysimeters. And during these 3 years, I think the number of pore volumes of the, I mean, if you take this 170 centimeters soil depth and look to the amount of rainfall that percolated through, you will find it was less than a pore volume. While in the columns, we had, as you have seen, we had 6 pore volumes a day. Mm-hmm. So we had many more pore volumes. So we would, if we would have run this experiment over many more years, then I am not sure that the iron will stay there and that the antimony will stay sorbed. That I think over decades, it might still be mobilized. So I'm not sure. But on the other hand, we will have accumulation of organic matter, as I said. This wasn't, and organic matter, of course, also sorbs antimony. So it could work. It is known from peat bog investigations in Scotland, actually. Bill Shottick, I know he did some work, and maybe also Margaret, you were on the paper, right? You were on the paper I've seen where they investigated the option of antimony by organic matter. Yes. So this could replace the dissolved oxides in a peatland situation. But I think in general, you have, at least in Switzerland, you have rarely these conditions where you would have a safe containment of the antimony. What they usually do is what you see also on the front picture, remove the soil, exchange it, or and then put it into a waste disposal, clean it by a lot of done by this Chur soil. The heavily contaminated soil was washed, was put into a soil washing facility and was washed, especially the gravel below the topsoil, and we used for construction purposes. And part of the soil, as I said, can be reduced, which is only low level contaminated, with the results. We have the antimony is not the main problem. If the lead is not a problem, I would say you can use it for purposes like roadside construction, landscaping, and so where lead is no problem, then antimony will also not be the problem.
Okay, so is there other questions from those of you who are online? I'm not seeing anything typed in. I just wondered if you'd like to comment on how you do going from the experiments to the laboratory analysis, because you obviously have to preserve the oxidation states. Are there any special steps that you need to take, any special difficulties that you find when you're working with antimony?
Yes, I mean, this work with the sterile condition, they work in the glove box, but of course, the samples taken from the field, you could not preserve them sterile, so they were immediately. I mean, one thing was I have to look actually into the PhD thesis of Kirsten to be really sure, but she made sure that they were under a nitrogen atmosphere and that they were processed as quickly as possible. Mm-hmm. So they were brought into the lab, and also all the samples done in the column experiment were analyzed within 1 hour from the column experiments. So time is really an important issue in it.
Yes, yes, that's certainly something we faced as well. The 1 hour period really is very important.
For the metals, we usually also acidify the samples. Mm-hmm.
Are there any further questions? I'm just leafing back through my notes here, and we touched a little bit there on organic matter, and there is a bit of debate in the literature as to whether organic matter is important, with some researchers thinking that iron oxides are much more important, and really organic matter is not involved at all. Do you have thoughts on that? I mean, certainly our work suggests that there is a role for organic matter.
I think there's an important role for organic matter, and one role is as a potential sorbent in soils where you have like in soils where you have a lot of organic matter, and depends also on the pH. And I know from Matt Tiger in New England University of Armidale in Australia, you know, a group of Sue Wilson, they in his soils or their soils, they found that organic matter or clay is an important part, probably because they have a lot of organic matter in their wetlands and not so many oxides. I think where we have a lot of oxides, the organic matter plays only a minor role. But where it plays a role is when you have dissolved organic matter carbon. We found there's quite a competition. Kerstin won other experiments where she did competition experiments with humic matter and with citrate on Goethite, and she found quite a competition effect. So, and we also found the seasonality. Actually, I didn't mention it in our drained lysimeters, but there was much. There was more in the effluents than in summer than in winter, which showed quite a close correlation. So 60% of that variance was explained by the DOC concentration in the effluent. So there is competition between organic anions and antimonate. So I think the organic matter plays more role than it came out from my talk.
Yes, yes. Yeah, that's very interesting what you just said there. I see another couple of questions coming in. There's one thing that I'll finish off with that Doug has asked me to mention. But first of all, there's a question from for management attempts, if we keep the soils waterlogged to immobilize antimony, how would lead behaved? Is that immobilized as well?
Of course, lead is then also mobilized. So I think that's one of the problems you face. And unless you decrease the redox potential even low and have sulfur in your system and you get precipitation of sulfide, lead sulfide, I think you may then have a lead mobilization problem. But I mean, also lead is strongly sorbed by organic matter. So in the case of these wetland soils, I'm thinking where you have like or peat soils, I think then the organic matter would also take care of the lead.
Yes.
But we have certainly to take this into consideration, that we do not only focus on antimony.
Yes, yes. I think that's the thing when there's multiple contaminants. But yeah, we certainly also found that lead is very strongly immobilized by organic matter in peatlands, and there's a strong correlation between the atmospheric records and the deposition records when we're in a purely organic system. There's no post-depositional movement.
One point, lead speciates in solution as a cation, right? Also. And we have the antimony, as antimonate is an anion. There we have actually a problem because sometimes people said, okay, let's just increase pH, let's lime the soil, and then we immobilize the lead. But with the antimony, it would have the opposite effect. So
yes.
What we also see in some experiments have seen that we mobilized antimonate when we immobilized lead. So there we are really in a what they call catch-22 situation.
Yes, yes, absolutely. Yeah, tricky issues. Keeping all our potentially toxic elements under control at the same time. Okay, so I'm not seeing any other questions. One thing I would like to say that Doug wants me to mention is that all of the lectures that have been given so far are now online, and today's has been recorded as well. So if you want to go back over anything that you've heard today, or if you want to listen to ones that you've missed, then you can just click on them on the IIES website. I'd just like to say thank you again, Rainer, for an excellent discussion as well as a fantastic lecture. And I think we log off now, and if there's MD who wants to continue discussing, then they're welcome to do so. So thank you very much.
Thank you very much too. Thank you.
Okay.
Bye-bye.
Bye-bye.